All right. I don't know if anyone's going to show up. Says that we are live. I'm got to wait here, I guess, for it to actually go live. Got 9 waiting. There we are. DJB says hola. And very respectful dialogue. No ad hominem. That's true. I held back. Oh, great. Thanks. Appreciate that. Base theory. Yeah, that was all right. I mean, a little frustrating because we're talking about completely different things. We got Bubblegum Gun wants to come in here and debate. Thanks for the sub, Guitar Dog. Appreciate that. Okay, we got 9 in here. So, okay, yeah, you said he did a horrible job. I won't say horrible. I think that he comes from at this topic from a different perspective. And the thing is, I am a biologist, and yes, so T-Rex did a horrible job. All right, I'm going to put that up there. I think if it was on a slightly different topic, it would have been harder for me. I am pretty good at these debate things, and I end up kind of pushing towards topics that I know better. And I assume that the creationists should know some about these things. It doesn't always work that way, obviously. There we are. Good job. Thank you, James. Oh, and then Bubblegum's saying, get out of here. All right. I don't know what's going on there. Yeah, I know. Skeptic Alan, I totally agree. It's frustrating to hear attacks on dating methods. If you're familiar with it, can you give the elevator speech supporting dating methods? I actually put together a few slides. I mean, geez, I'm a professor, right? I can't talk without having some slides. So I could actually show a couple of the slides that I have put together on dating methods. And there's a really great paper. I forgot to put the link in it because I was just throwing it up at the very end. But let me present. Actually, let's do present slides. Okay, that's not going to work. I'm using StreamYard to do this. All right, I'll just share my screen. And we're going to share this screen. Okay, so yes, we can see this. And I can find where this picture came from. I didn't get into it too much because I know that TRock is pretty strong on some of them. I mean, stronger than I would be able to handle on dating methods and radiometric dating, since that's not what I work on. I'm obviously familiar with sort of the basic core arguments, but I don't think I would feel super comfortable dating it. But when he says like carbon-14 dating is good only up to 3,000 years, that's just complete garbage. We can look at that here. So we have to make some estimates of the accumulation of atmospheric carbon-14. Carbon-14 arises from radiation from the sun acting on carbon molecules that are in the atmosphere, and it creates radioactive carbon, and that's the source of radioactive carbon-14, and then it decays. Okay, and so there's going to be a window of the amount of carbon-14 that you would expect to see, and obviously that window gets a little bit larger as the farther back in time that you go, right? And because that can vary to some degree, although it's not substantially varied. And this is the window that they're talking about here. Now, what we can do is look at the carbon-14 dating and look at the ratio of carbon-14 or the loss of carbon-14 relative to the carbon derivatives. I think it's carbon-12 that it decreases to. And this is the window that we would expect like data to fall into if our understanding of carbon-14 values to be conventionally true is correct. So it should be like above, it should be within these two purple lines. And here's 3,000 years old. And so those 3,000 years, so if this is 10,000, here's 3,000. 3,000 years ago, you still have around 75% of the original carbon. That's still plenty of carbon to work from. The half-life of carbon-14 is on the order of around 8,000 years, it looks like. And so that means that like at 10,000 years, you're still working with plenty of detectable carbon-14. Okay. Other methods that you can use. We can use dendrochronology. So I thought this was a really nice way of showing dendrochronology. So trees create tree rings. The rings vary in their size. And from an existing tree, you're going to have certain rings, like you take a core out of a tree and you look at the different bands, because like there might be a lot more rain in one year or not so much rain in another. And then this can lead. So, but the thing is, like, we can also get older wood, like say wood from an older home, and look at the rings in an area there, and you can match some of the rings. So from an existing tree, like the bands that'll be towards the center, this will be like the youngest. This is when the tree was really, really tiny, and this is when it was much older. And you can match the bands to other wood sources. So they're all going to match to some degree because they're all getting the same amount of water. And then you can take that older wood sources and compare it to even older wood sources. And you can keep going down further and further and further. And in some areas, you can get overlap of a dendrochronological sequence that goes down to like 14,000 years back, dating year by year by year. Okay. Now, carbon-14 dating relative to tree ring count. So if you can go through so this is like actual data, a tree ring where they went through and did carbon-14 dating on individual rings to determine how much carbon-14 is in there, right? Carbon-14 is going to be incorporated at the time at which it's alive. And then that ring, will go towards the center, and then the carbon will start to degrade some. And then you're going to create a new ring outside of that. And of course, like older trees, like from 8,000 years ago, the carbon-14 should degrade over a period of time. And you can see that the tree rings match. If you date the carbon-14 in the tree rings, it goes right within that window that you would expect carbon-14 to actually be working. You can also do this with varves. So varves are, thin layers of sediment that's laid down in an annual basis, because certain times of year, you're going to get certain kinds of sediments that'll have carbon aspects to it. Certain like sediments or organic material that gets laid down and then it goes away. And so each of these little bands is like a year. So there's an annual cycle to this. So varves is another way of keeping track. And here's more data from looking at doing carbon-14 on varve data. So taken from varves. And if you match varves to tree rings and you look at the annual layers over the overlap for these two data sets, and you look at the carbon-14, and it is perfect. It is damn near perfect. You couldn't ask for better data. Now, TRock's talking about like, oh, you have to like calibrate one against the other. I mean, we don't have to calibrate these. Like, we have the rings. We're just counting the amount of carbon that's in each of the layers, or of the tree, or in each layer of the varve, and you can see that it's like perfect. It's damn near perfect. So yeah, maybe took a little bit long, but let me see here. Oh yeah, hide current comment. All right, so there's a lot of stuff going on here. As I was talking. Oh, Dalmatian evolution. Oh yeah. So did you guys say that last time and then last debate? All right, bubblegum. Do I need to kick you out? Don't be an ass. Right. So we have an argument going on in the chat. Okay, let's take this one. Food is literally the building material of what we are. To say it doesn't affect DNA is absurd. What I'm saying is that it doesn't cause mutations. When you eat food, it doesn't cause mutations that are going to be passed down from generation to generation. Yes, of course, you have to eat food in order to, say, have cell proliferation. And then you make a new cell, you have to make new DNA. Food, when you eat it, it's going to affect the expression of genes. Absolutely. But that doesn't mean it causes mutations in the DNA. Yes, the food is primarily turned into glucose. That's exactly right. The food is turned into glucose, which is what the body needs to process and create energy. Glucose is then processed by the mitochondria to create ATP. So, all right, bubblegum, don't be. I'm gonna. I don't want to block people if I have to, but now I know who you are. Okay. Honestly, I don't know who you are. Did you ask the Dalmatian question last time? Is that what that was? About dogs and how like Dalmatians, like if we know how Dalmatians come about or whatever. I would agree with this. If you can't be polite, you're going to get booted. All right. I I did a debate last October. I was Aaron Ra's partner. We debated Sal Cordova and Cindy Lincoln. That debate went about how this one went, I feel like. And there was someone who asked some question about Dalmatians or something in the chat. And so I'm just connecting the two. I don't know if that was you. All right. I would appreciate that. He thinks evolution is all devolution and that all mutations are harmful. He thinks a beneficial mutation is impossible. Cool. Well, why don't we talk about that? Let's go back to the ancient DNA stuff. Okay. So I'm going to, oh, wait. Oops. I should have kept sharing. All right. Neanderthals and humans had approximately the same brain size. And in fact, Neanderthals were probably a little bit larger, it seems. But the thing is, modern humans had a larger frontal and prefrontal cortex, okay, cortices. It's slightly larger in humans than it is in Neanderthals, despite the fact that Neanderthals probably had a slightly larger brain. But they had a smaller frontal and prefrontal cortex. That is important because those cortices are really important for logic and reasoning, okay? The other thing that we know is that Neanderthal genome had a TKTL1 in their gene, had a lysine at one particular spot, but humans have an arginine. Okay, so we know this from ancient DNA. And this is the paper that describes this. The human TKTL1 implies greater neurogenesis in the frontal neocortex of modern humans than Neanderthals. We can look at the DNA. I made these images myself. I went ahead and blasted the human TKTL1 gene against the pan gene, okay? And what we can see is that, so pan is chimpanzees, pan troglodytes. And what we can see is that they are almost completely identical except for this mutation, where there's, in humans, there's an arginine represented with an R, and in chimps, there's a lysine. And what this means is that Neanderthals had mutate. Well, they likely had the. So if we accept that mutations occur, and if you look at 3 different organisms, chimps, humans, and Neanderthals, and we're talking about this one feature, in this one feature, it turns out that Neanderthal TKTL1 is more similar to chimp TKTL1 than it is to human TKTL1. So between Neanderthals and humans, there has been a mutation that is specific to modern Homo sapiens and not in Neanderthals. Neanderthals were more chimp-like in this one respect. Okay. So this means that must have been a mutation in TKTL1 in the human lineage that was after humans and Neanderthals split. Okay, so a mutation, big deal, right? So the thing is, mutation, neurogenesis is it happens in the inside of the brain. Brains grow from the inside out. So you have neuroprogenitor cells that derive. That's what these NPCs are. They are born in the cells. These cells proliferate, and then they divide, and then they differentiate into neurons. This cell population is critical for the expansion and enlargement of the cortex. Okay, humans have relatively high rates of neuroproliferation that occurs in the cortex. Part of the reason why we have such a big cortex. Neuroprogenitor cells have this very specific morphology. So this is something that I actually study in my lab. This is why I have this figure. So neuroprogenitor cells, their cell bodies live here along the ventricle, and they have these long processes that extend up to the surface of the brain where they have these end feet. Okay. Now, in this figure, this study that just came out, this is in 2022 from Savante Pabo's lab. What they did is that they had mutant mice that they made with human TKTL1, Neanderthal TKTL1, or controls. So they changed mice to either have the human version or the Neanderthal version. Now, the Neanderthal version here is known as A for being ape because it's more like chimpanzees and gorillas. Okay. So if you put the human version in there, you get a lot more of these neuroprogenitor cells, these mitotic basal radial glial cells is another name for neuroprogenitor cells. And you can see that the increase in the percent of the population that has more of these, it's like tripling. So we have a single mutation that's occurred, and it allows for this massive expansion in the neuroprogenitor pool. And the only thing that's different between this and this mouse and this mouse, is that this has the Neanderthal version and this has the human version with a single point mutation, one mutation leading to a change in a single amino acid being changed. Okay. This leads to even larger neocortices in ferrets. They did this in ferrets because mice don't really have like folding of their brains, but ferrets do. So they did the same thing in ferrets, and ferrets with the human TKTL1 version have a larger gyrus size. So they have larger frontal cortices if they have this single mutation. So a single mutation can lead to substantial change in overall morphology in something as complex as the brain. There you go. Is there any evidence of increase in brain size function of modern humans? Well, let's see. All right, wait, let me go back and look at what's going on here. All right, I haven't booted anyone yet, so play nice, everyone. All right. Increase in brain size and function in modern humans. So, like, you mean over the recent decades, or do you mean like over eons? Or we're talking about over thousands of years? Because obviously there's been an increase in brain size in across Homo, and certainly from Australopithecines to Homo. But if you're talking about Homo habilis to Homo erectus to Homo sapiens, yes, there's an increase in brain size. But if you're talking about Homo sapiens in particular, honestly, okay, there's not a whole lot of evidence of an increase in brain size because the earliest fossils that we have of Homo sapiens, I believe, they're still like pretty largely encephalated. There's some evidence to think that modern humans may have actually decreased in brain size, that Cro-Magnon man, modern human Homo sapien skulls from around 50,000 years ago, there's a couple of studies that indicate that they actually had larger cranial capacity than modern humans. So if anything, there's actually been a little bit of a decrease, and some estimates are that it might be as much as 10% of a decrease in brain size that's happened in modern humans, which is kind of wild to think about. And there's some data, or there's some theorizing at least, about why that may have happened, which is a self-domestication phenomenon. Nice. So yeah, a little bit about what I do. Like, of course, I like arguing about creationism and evolution. I like arguing about other things. I haven't updated my channel in like a month, but I'm picking it up again. Uh, um, I do make videos on neuroscience and make videos on evolution, um, and and I'm also trying to make more videos about evolution and the brain. So if you're interested in the interface of those two things, that's something I'm interested in. So like for me, it's just got to be something I'm interested in. So I'll be doing more stuff on that. Yeah. Would it be neuron density more than brain size? It depends. So if you're talking about like the change in brain size in humans, well, we can't really say anything about what neuron density looked like in Cro-Magnon man, like from 50,000 years ago, because we don't have that soft tissue, obviously. Changes in brain size can obviously occur through differences in neuron density. So if neurons are packed more together, obviously the brain's going to be a little bit smaller. I would say, though, that in general, neuron density does not vary substantially, at least in the cortex, between, say, humans and non-human primates. The neuron density tends to be pretty similar. And so I would assume that, say, like humans from 50,000 years ago probably had the same neuron density, and they probably had even more neurons. So, do you let people in the hangout? Oh, to chat. Because Nestle, we've talked before. Yes. How can I do that? Yeah, because you want to chat. Let me see how I can do that. Can I just drag? No. Link to join the talk. Ah. Oh, all right. Now, help me out here. If I, okay, I got it. I think. I don't want to put the link on here if there's going to be a bunch of arguing and fighting. Okay, I'm not too sure I know your email, though. But sure, I'd be happy to chat. If you don't mind putting it here. Oh, yeah, that's right. Okay. All right, I'll just do that. And I can pick who joins. I am brave. But I don't have to accept people who join. But go ahead. We're going to see how this goes.
Hello, can you hear me?
I can. How
All
are
right,
you?
great.
Good seeing you again.
Doing great, thank you. I did not get to see the debate though. Sorry about that.
Oh, that's all right. I don't know if you missed much. Yeah, so it was interesting. I I think TRock needs to learn more biology. Um, he had some pretty wild ideas around mutation, and that like eating sugar somehow causes mutation, and that's how people get diabetes, and that's why like people diabetes is heritable, and if you had a family that's just eating sugar, they're going to get a mutation for diabetes.
Okay.
I kind of checked out at that point. It was like, you are not following this. Let's see. All right. Yes. I'm going to add you in there too. All right. So we have base theory in here. Hello.
Hello.
Hey, how's it going?
Good. Yeah, I did catch the debate. My name's
Okay.
Grayson, and actually, I'm going to be debating TRock in like a month or something on the age of the Earth. So I figured, this could be good oppo research, kind of see, because I haven't seen him in a debate before. So let me just say that seeing your debate with him tonight definitely gave me a lot of confidence going forward for our upcoming debate.
Good. Yeah. I do think that he's a little solider on age of Earth issues.
Yeah, so that's the topic that I'm going to be discussing with him is age of the Earth. So he's listed out the kind of points he wants to talk about. Okay. But after hearing, muddy water causes mutations, and he didn't seem to understand that like genes are located in the nucleus. And
we can't have the conversation.
Don't go to the nucleus.
Yes. Right. And it's like, I can't explain this to you right now. So how will we even have a conversation about this issue? And so all that stuff Emma was talking about, I mean, it felt like he kind of understood it, but when he was thinking like that, just the stuff that you eat can just affect your genes and cause mutations that'll be passed on to the next generation, I was just like, okay, we're crossing roads here, and
like you cannot even have a conversation at that point. It's like you're not it sounds condescending, but it's true. Like if one of the persons doesn't know the subject, then you cannot talk about it.
It's true. Yes.
Yeah.
Especially in a debate. Like I'm fine explaining something to someone. I'm happy to do that. Like if we're just talking now, of course, that means that I'm going into lecture mode and I'm going to be telling them DNA is located in the nucleus. Things you eat don't have mutagens in them. There's a list of chemicals that are mutagens, and it's not that long of, I mean, sure, there's plenty of chemicals that cause mutations, but we're not eating that. Okay. Right. That would be a big problem. So,
and right from the start, like your intro, right, was on topic with evolution creation. You were actually talking about facets of creation, talking about facets of evolution. And then his intro was almost nothing about the theory of evolution.
I've seen him do that intro before. Okay. So if you want to look at a little bit more, you can go to like basically in YouTube, you'd have to Google search Standing for Truth and TRock, or just go to TRock's page and load in like every video, or sorry, not TRock, Standing the Truth page, load in every video and then search for TRock, because like the search algorithm in YouTube also sucks. It's kind of lame. But he's done it a bunch of times. And he's got debates specifically on age of the Earth. So if you haven't seen that yet, definitely do that.
Yeah, and he already told me some of the arguments that he was going to be making, like talking about erosion rates being too fast, that all the continents would be worn away in 1 million years. He talked about, the moon is moving away from us.
Yep.
Like, and all these things that are fairly easy to address and debunk. So I'm looking forward to the conversation, honestly, because like he's kind of I don't know if Dunning-Kruger would be the right word, but he definitely he reads enough to where he confirms his own bias going in, and then he thinks that he has the whole story. And he was actually calling you out, saying like, Dr. Thompson, I don't think that you've done the reading on this. It's like, Have you,
Did you
T-Rock? I'm
debate?
pretty sure.
Yes, when he's
Did you
telling me
debate
like
standing? Did you debate standing for truth or someone associated with standing for?
it's someone associated with standing for truth.
Right.
So it's a guy named TRock. He goes by TRock. He's kind of an acolyte of Standing for Truth. So Standing for Truth hosts a lot of debates now. It seems anywhere he brings on.
It doesn't really surprise me that someone who believes that like eating food mutates your DNA associates with Standing for Truth. The people who he associates with, they are the epitome of Dunning-Kruger's. Like they think they know better than anybody, but no, they don't.
Yep. No. And I think you're right, base theory, that there's definitely a Dunning-Kruger effect going on to some degree. He's a knowledgeable guy, I'm sure, in the stuff.
That was me. There was also the other guy. That was me.
yeah. Base theory. Yeah.
No, Neslick.
Yes. No, you're right. Neslick was. You were saying that, but you were base theory. You go by base theory.
You can call me Grayson.
Grayson. All right. Yeah. So Grayson, you were talking about that. Like, is it a Dunning-Kruger effect? And for sure, I think so.
Right.
Yes. Because I think that he's probably a knowledgeable guy in the things that he knows, but for the stuff that he doesn't know, it's clearly he's just feeding on young Earth creationists crap and doesn't understand the nuances of any of the arguments that he's actually reading. And so, yeah, for him to talk about the lunar, the distancing of the moon over time, I mean, that's like Kent Hovind garbage.
Well, he draws on Kent Hovind. He name-dropped a couple creationists in your debate tonight. He mentioned Nathaniel Jeanson, which I don't know if you've done any work on Nathaniel Jeanson. It's hilarious. He's written a book, Traced, where he uses genetics to try to prove like Noah and Ham and Japheth and everything. And by his own calculations, using his own mutation rates that he gets from like pedigree studies and things like that, he calculates that the R1b haplogroup that most Europeans are members of did not come into existence until like the 1500s or like the 1700s or something, using his numbers in the book. And then you can just go and look at, ancient DNA studies of Romans and you can find R1b. So it's very easy to debunk.
Right.
But he literally is a PhD from Harvard and he's pushing this. I mean, it's pretty insane the lengths that you'll go once you get hired by one of these creationist institutes.
So one thing that I did with the bear thing, so I had gone back and looked at a couple of TRock's debates, and I saw that he had brought up bears as an example of a kind, and I thought, okay, I'm going to invest a little bit of time, creating a couple of slides on bears, and because I know that there are two outgroups in bears, right? We've got pandas, which are substantial outgroups. So I knew that their genetic distance is a long distance away from the rest of the bears. And then I knew there was another one. I don't know all the bears. I don't work on bears, but then I looked into it, like the spectacled bear. Okay, that's different. And then you got a bunch that are clustered together, right? So the thing is, like, I find that evolutionists, I hate the term evolutionist, people who are arguing in favor of evolution, because it makes it sound like, oh, it's like a highfalutin, but we're believers in something, because that's not the case. People who are supporting and defending evolution, they end up doing too much of the defense. And what you need to do is take some of the conclusions that they have from their own model and follow the data and walk them through how stupid it is. So if he agrees that there's random mutation that occurs and that random mutation is going to insert into the genome, you can use that as a somewhat of a rough clock at the very, very least. And obviously, like, we have sophisticated methods for doing that and comparing genomes and how they're different from each other. If you're talking within a kind, you can still do that molecular clock dating within a kind. But the thing is, like, that what it clearly shows is that they didn't all diverge all at the same time. That's what I was trying to show. That if you then date now to when the last common ancestor was between pandas and the rest of bears, I bring it up. We're stuck with 4,000 years, right?
I thought that was like a really good approach that you took, and I've had debates with Standing for Truth before about subjects like this, especially when you brought up Neanderthals. It's a subject that Standing and I have gone back and forth on a lot.
I'm sure.
And basically how he would explain this chart. Obviously, you saw it tonight where TRock just moves the goalposts.
Yes.
He'll just say,
now we have more
if there are
kinds.
3 bear kinds, if there are 8 bear kinds, no skin off my bones. Like he's willing to just shift the goalposts as much as you pointed out.
Exactly.
But there's another approach where they'll just say, oh, well, like the one of these species is a hypermutator. Their mutation rate is so fast that even though they're only hundreds of years, your molecular clock looks like it's thousands of years because they acquire mutations so much faster. So those are the 2 main like rebuttals that I've come against by trying to make these arguments where, it's just they just kind of retreat and then move the goalpost to something else.
Move the goalpost and retreat. And it's there's an argument that it's the Moat and Bailey type of argumentation where
Bailey.
Yes. So like the moat is the sort of the field. So this comes from a form of medieval castles, the way they were designed. A given sort of little settlement would have this like large field where you have like agriculture, but it would be like a ditch around it so that if like some attackers were coming, they might be stopped by the ditch and you can kind of push them away. But then you would have this fortress called the Bailey where everyone could, if they were getting really overwhelmed, they could retreat into the Bailey and just kind of throw rocks over the Bailey and over the edge of the Bailey and knock down the intruders. Now,
so like a reverse straw man, they misrepresent their own position in order to defend the real position, basically.
Exactly. They will retreat to the easily defended Bailey and just argue from there. And so now I think one strategy that like we have to take is that you got to just keep them on the moat. Like you can't get them into a Bailey. And so like when I argue with like Sal Cordova and he wants to talk about abiogenesis and like the protein families and stuff, like I'm not going to go there. Like I already know that there's but I can use the stuff that I know he's going to talk about to get him back on the moat and to talk about recent evolution and what we actually know about evolution. And so I did that in a conversation with him when he wanted to talk about potassium ion channels and like the family of potassium ion channels. How can you explain the origin of the potassium ion channel? And it's like, I'm not going to explain the origin of the potassium ion channel. However, I'm going to use the potassium ion channel to describe a couple of important concepts about evolution to you. And now you will not have, I'm going to set up an argument that you cannot possibly defend as a creationist. I had this discussion with you. I don't know if you guys have seen it, but it's a pretty great discussion. And he ended up at the end, he was like, I forgot to tell you, I have a meeting. I have to go. And he just left after I like laid out this whole argument. And he had no explanation other than miracle.
I want to check that out. Is that on? Was that what you were talking about on Modern Day Debates with like the 2v2, or was that a one-on-one with you and Sal?
That was just one-on-one with me and Sal. Yes,
I'm gonna have to check that out for sure.
I've hosted him a few times on my channel just to have conversations. But they're debates, right? Like,
so this was on your channel?
Yes, so this is on my channel. Yeah,
gotcha. I'll definitely check that out then.
Yeah, check it out.
Yeah, this is one of the major problems I have with these people. Like in any field or any theory of science, there are always gaps because scientists are human. They don't know everything, so there are always mysteries to be just to be solved. So it's easy to attack even a well-established scientific discipline, but it's much more difficult to come up with an alternative. And when you ask them, oh, okay, we don't know the origins of for example, the potassium ion channels. Exactly. What is your solution? Miracles. Okay.
Right.
No thanks. Right.
But as long as you can get them to admit that and then stay on that and say, okay, I guess we're not talking about science, are we? We're talking about theology. You're not talking about actual scientific methods. Because if it was an event that actually occurred by an intelligent designer, there should be things that we should expect from that. Like there should be like traces of a designer and how a designer would go about doing that. Explain to me even how the miracle would have occurred to create this potassium ion channel. And so if they're going to talk about potassium ion channels being in separate kinds, right? So why are certain potassium ion channels similar across similar species and how are they different across disparate species? And that's actually where I get into. I actually don't even want to ruin the surprise, because I set him up with this thing where, and it's just like, I mean, I don't want to sound like I'm bragging, but it really was quite beautiful the way I set it up. And I can understand why he ran away.
My instincts from all the debates that I've had with creationists would be that they would bring up something like, it's not common ancestry, it's common design.
Sure,
I hear that all the time.
But like the way I set it up, because I'll give a little bit of a hint, it involves some convergent evolution, and I did a lot of work to do this. And as you know, I don't want to say much more than that. It's a thing of beauty.
One of my personal favorites that I'll respond to whenever I get it's just common design, not common ancestry. I always like asking about I ask them to make predictions based on common design. So I'll give them like 3 animals, and I'll say, okay, which 2 are the ones that are the most closely related genetically, and which one is the one that's the most different from them, based on a prediction that you would make using common design to make these predictions. Right. So the one I really like is I asked them to compare goldfish, sharks, and humans, and say, okay, so is a goldfish more genetically similar to a shark or to a human? Because common design, obviously, the goldfish should be more similar to the shark, but in actuality, they're more similar to humans than to sharks.
Exactly.
Because, they're bony.
That's a great example.
My favorite example would be like comparing, for example, sharks with whales and humans, and look at the underlying structure of whale flippers. It's basically a human hand almost. Like it's the same digits in the flipper basically. So why do we have the same quote unquote design as whale flippers then, and instead of whales having shark flippers? Like why?
Right. So I'll spoil it a little bit. So what I ended up doing was, first of all, talking about that there's a family of potassium ion channels. Because, Sal talks about like explain the origin of the potassium ion channel. And in fact, that there's actually quite a bit of different kinds of potassium ion channels. And it's a lot. Like there's probably around 80 potassium ion channels in the human genome. And there's only about 40 that are really kind of described to any degree. So there's a bunch that we don't even really know much about. Okay, so already it's like you're dealing with stuff on the edge of what we know for the most part. But we do know a lot about many of them. And the thing is, of course, you can just compare sequences, and that's pretty easy. So we already know that like there's certain potassium ion channels that are more closely related to others. And that can lead to like a tree that exists within humans when you compare like the human KV 1.5 to the human KV. KV is the potassium ion channel. Like this is a voltage gated potassium ion channel. To the 1.8, like they're more similar than either of them are to like the KV 1.1, for instance. But this is all within humans, right? And this is just one subset of the family of potassium ion channels. And so what I ended up doing is comparing like, so you guys might recognize this guy. The humans and chimps are going to be more similar, and then mice and rats are going to be similar to each other. And if you compare the human KV 2.1 to like others, so humans and chimps are identical, but if you compare them to rodents, they're different. And if you compare the mouse KV 2.1 to rats, like they're a little bit more different because rats and mice are actually a bit more different. And then you compare them to humans, obviously it's even more so, right? This is all pretty basic stuff. And they just walk them through this. But then like, okay, we don't have to do it with just 2.1. We can pick any of them. We can pick 6.2. And so, humans are more similar to chimps than they are to rodents, and vice versa. But then, of course, you can also look within just an individual like KV channel to other KV channels. So KV 1.1 is more similar to KV 1.2 than it is to say like 6.1 or 6.2. But this is within humans, right? So there's a phylogenetic tree even within humans as well. And so then I ended up trying to get them to talk about other species, so outside of just humans and chimps, and we ended up focusing on myomorpha, and which are the suborder of rodents, with the 1,524 different species. And we talked about different rodents, and that's where I'm going to stop. So like you gotta go look, but we ended up looking at rodents and then other things that aren't exactly rodents but look a lot like rodents, and comparing the different KV channels. And so that's where convergent evolution was a really great example of like, okay, you don't have a good explanation for this, because he would, what Sal would say, or other creationists would say, is like, well, of course mice and rats have similar DNA, because a designer would give them rodent-like potassium ion channels to do rodent-like stuff, right? That's why they would have that. But we can find other animals that look like rodents but aren't, and do they have rodent-like DNA or not? And yeah.
Well, that's where they move the goalpost on you, and
that's where
they have to come up with this story, right? Because
sure,
in their worldview, there's no such thing as like non-constrained DNA sequences. Like every part of the genome has to be functional, and like they cannot, like for them, they're not willing to budge on that. So even if there is no function that can be established, or even if like there is, like we can show that these different types of myomorphs are under similar environments, under similar pressures, like they have common designs, they should have similar DNA, and then when they don't, they'll just have to fall back, move the goalpost, and say, oh well, I'm sure like the differences are functional for their niches, and we just don't know enough to explain it yet, but I'm sure it's 100% all functional and designed, and like they just have to fall back on believing that, because they can't demonstrate it.
Now, okay, I'm going to test you guys.
It's also important to know that those excuses are always made on the spot after the fact, that they don't predict anything of this.
That's right. Yeah, they don't have to actually make predictions. But the thing is, like, we have the power to force them to make those predictions. Like, we can tell them, isn't this a prediction of your theory? And can you explain it? And if they can't explain it, that, there's some value in seeing them move the goalposts, as you saw the TRock did today. Like, I actually clipped a video of him talking about, like, yeah, there was one bear kind on the ark, and that, like, that's where we get 8 species of bears. And I didn't play it because I was like, okay, well, that would be a little bit much for me to, like, oh yeah, well, you said there was only one bear kind, but he already was like, I'm fine with slipping. It's okay, fine. You're just shifting the goalposts. Fine. Like, all right. So here's a little bit of a quiz for you guys. So I say carnivores look very similar, right? Do you see anything weird about the images?
There is. Huh? One is not like the other. Oh, yeah.
There's a couple that are not like
that. There's a couple
two of them are not like the other
on your list there that are not carnivorans.
Do you know what they are?
Yeah.
The Tasmanian tiger, and then the Tasmanian devil there.
That's right. Exactly.
Correct. Yeah.
So, but Sal doesn't know that. So, like,
Oh, really? Didn't he guess the?
No. I went through it pretty quickly. So this is the trick that I did. So this is also the marsupial mole compared to other moles. And then these two guys are marsupials too. They're opossums. They're South American opossums.
Although the one on the bottom right is the Monito del Monte. It's like a separate lineage from the even from the opossums.
Yes, it is. That's right. Yes. However, it's still a marsupial.
Yeah, it's still
so therefore it's different enough. And so what I was able to do,
exactly the one in South America that's the most closely related to the Australian marsupials, the Monito del Monte.
Oh, okay, that's interesting. Yeah.
So you have the marsupials, like you have one lineage that goes to the opossums of like the one in North America, and then you have then the one branch goes to the other marsupials, and then you have another branch I think that's the Monito del Monte, and then sister to that is the Australian marsupials, if I'm not mistaken.
Interesting.
Yeah.
Yes. No, I know that the Australian marsupials are a separate lineage from like the South American ones. I didn't realize that the Monte del Monte is
closer
separate
to the Australian. Yes. Yeah.
Yes. That's super interesting. And then no wonder that it's been sequenced. I was thinking, like, why did they sequence both of these guys?
It's actually interesting. The Monito del Monte has fossil relatives that are found in Antarctica. And the thinking is that the Monito del Monte may have been a back migration. Like the marsupials began in the Americas, and then they migrated via Antarctica to Australia. And perhaps some went back to like Monito del Monte went back to the Americas
before they separated. Oh, that's super interesting. I didn't realize that. I didn't look into it that closely, but that makes a lot of sense. And that would also explain why, like, if you take these potassium ion channels and you compare like the mouse potassium ion channel to the other myomorphs, that they have very high homology, And if you compare it even to humans, obviously it's very high. But if you compare them to these rodent-like marsupials, it's actually quite low. And if you do this, you can do this for all of them, like the KV 2.1, the 6.2. And Sal's explanation for this was a miracle. That's all he could say.
I would have imagined.
It's funny how the same explanation explains everything.
Yeah, I would imagine that they would say, oh, well, like the human potassium ion has to function on human potassium, and then all these others, it's species-specific potassium. That's why they're different for each species.
But I had already walked them through that these are rodent-like animals living rodent-like lifestyles. And
I don't think it's a tenable position. I just think that that's what they would fall back
I'm sure
to, saying. There's something unique about each of these species on an actual level of the potassium ions. There's got to be something unique because that's how it has to be different. That has to be the explanation. Like they just fall back on that.
No, and you could see his wheels turning, and that's what he was looking for, but it didn't happen. So
also, if I'm not mistaken, the potassium ion channel is. Maybe I'm wrong, but I think it's also homologous to a sub part of the ATP synthase. Like the ATP synthase is composed of like two major parts: the one that's in the membrane, and the one that's like sticks out the head of where the ATP synthesis happens. So I think the membrane part is like an ion channel, homologous to an ion channel.
Yeah, that's right. Yes. And then the intermembrane part actually does a bunch of different functions. There's some that are G protein coupled even, or they affect G protein coupled mechanisms.
G protein couples are really, really ancient family of proteins, so super family. Even.
Right.
Yeah.
So
like some G coupled proteins, they function like the opsins in your eye are G coupled proteins, if I'm not mistaken, and also have I think so, yeah. And also the like few proteins are also involved in the sense of smell. Like there are many different sensory proteins that are descendant from this.
From G protein coupled, that family. That doesn't surprise me because there's like hundreds and hundreds of different G protein coupled receptors. Yeah, that's right. All right.
One thing that's really interesting about your sense of smell is that we used to think that it was dependent on the shape of the molecules interacting with like the receptors in your nose, and that was what determines the smell is the shape of molecules. But that's not actually the case. Like you can have two molecules that are different shapes that have the same smell, or two molecules that are the same shape that have different smells. So what they actually figured out it was doing is it basically, like when it binds with the receptor is actually responding to like the resonant frequencies of the bonds within that molecule and not the overall shape.
Oh, that's interesting.
Yeah, it's all about the types of bonds present.
Yeah, and olfactory receptors, they can be quite promiscuous. Like they will be sensitive. They'll have high sensitivity to certain kinds of chemicals, and then but relatively low sensitivity to others. But that doesn't mean that they're not sensitive at all. And in fact, like when we have a sense of smell, it is the summing of the activity of all these different kinds of receptors, and then it being transported through the olfactory bulb, through the glomeruli, where it's a super beautiful system. I actually even have a video on my channel talking about olfaction and smell. Right. And even a neuroplasticity one related to smell. But then that's what allows us to have recognition of smell in the cortex. Yeah, I didn't know that about the resonance part. I assumed that, I knew that the chemicals could be quite different, but I just assumed like, okay, well, there's just enough interaction, energy here for some sort of binding to happen so that the receptor gets activated.
It was a relatively newer discovery.
That's cool.
I also mentioned something else about the olfactory receptors. Like again, there is in whales, as you would imagine, they are descended from terrestrial mammals, so they also inherited olfactory receptors. But there is like I think the odontocetes, the tooth whales, they in particular have they lost even the part of their brain involved in smell, so they are physically, like neurologically incapable of smell, and yet they still have the genes for olfactory receptors.
That's amazing. Right. That's very cool. Huh.
Well, of course, the creationists would say those genes are serving a purpose.
Some purpose.
They were designed that way. And even if we can't prove what the function is, there definitely has to be one.
Despite the fact that there's all kinds of olfactory receptor pseudogenes in primates, including humans.
Yeah.
But of course, must serve some sort of purpose.
There's also one thing to mention, like even if they have some additional function that they retained or newly involved, we can still recognize them as remnants of olfactory genes.
Sure.
Like it's an important point to remember that vestigial structures don't have to be non-functional. Like,
yes,
for example,
absolutely.
Yeah. Well, one of my favorite examples is the penguin flipper. Like it's a bird's wing, even though it cannot fly. It's still a wing, despite it being used for swimming.
Absolutely, yes, right, and it's still serving some sort of function. And so for when he, well, right, you didn't see the debate, but that was something that TRock brought up a bunch of times, but I didn't. It was always in response to some other question. I didn't actually deal with it, but yeah, right. There's nothing wrong with vestigial structures serving some additional purpose. Like otherwise it would be gone completely. Like it's just that if bodies are irreducibly complex, why can we be basically more or less just fine if you take out the appendix? And he referred to the appendix a couple of times, but I was like, okay, well, I'm just not going to deal with that at this point. He would end up saying something.
Yeah, or like gene knockout studies, that have knocked out like 50% of the genome of mouse and of a mouse, and they were just fine.
Yeah. That happens over and over again. Yeah. I mean, we know that there's huge redundancy in the genome within the body. And I plan on, but then a subtle change like this TKTL1 change can actually lead to substantial change in the function of an organ like the brain. And that's because bodies are very pleiotropic. We're not modular, and gene systems are very pleiotropic. Subtle changes in one can lead to substantial change in the function of others. And but it also means that because it's just so like gooey and stuck together, you can tolerate deficits in some aspects that will allow function to continue. And changing structure, you can't like really change one thing in an animal without affecting other things in the animal. We're an integrated system, and there's not it's very hard to point to examples of human made design.
Some parts can change. Some parts can change at different rates than others.
Sure, that's right too. Yeah.
I think a good example is like like one famous example in developmental biology is like the antler's head. Like you have what's called what's the term again? Like a change in developmental rates can lead to very drastic changes in the appearance of the end of the animal. Like I don't know, heteromorphosis, if I'm not mistaken.
Heteromorphosis. My brain is starting to fail on me.
Yeah.
Which actually might mean that I need to go because I've it's 11:15 here, so
yeah, I know that Bubblegum has been spazzing in the chat. He really wants to talk about dog evolution and Dalmatians. So if Bubblegum, if you want to come on my channel after this, we can talk to your heart's delight, and I can walk you through and hold your hand through dog evolution.
Yeah, do that. I don't feel like talking through it. I know from the last debate I spent a little bit of time looking at it, but yeah, okay, yeah, please handle that if you don't mind.
Yeah, there's a very simple paper where they do DNA analysis on like.
It's heterochrony. I meant heterochrony.
Heterochrony. Okay.
Yeah.
Cool. All right, guys, I am taking off. I am done. I am beat.
All right. Well, I'll watch that debate with you and Sal.
Yeah, do.
Yeah, it was nice meeting you, and
good meeting you. We'll see you around.
And
actually, if you don't mind sending in the private chat, could I find your channel someplace?
Yeah, so it's Based Theory, like my name on here.
Okay.
But I'll just send a link, I guess. But
no, I'll look it up. That's fine.
Yeah, so Based Theory. If you search like Based Theory after show, I should be probably the first one that shows up.
Good deal.
So yeah, thanks.
I'll see you then. All right. See you guys. Take care. And that is it for me. Adios, everyone. Bye-bye.